other helped us to prepare this talk. So thank you very much for all of you, especially Maru and Patti Salazar, that provided me a lot of photographs. So, Ricardo Tapia was born in Mexico City. He met Dr. Guillermo Maciel, who was a professor of biochemistry at the faculty, and worked also at the Institute of Biology doing research on the role of amino acids in the brain of different species. Ricardo accepted his invitation and he started to do research with Maciel, and from then on he decided to devote his career to science, leaving behind the medical practice. Ricardo published his first paper in 1962 when he was very young. He was only 22 years old. And in this work, they used this compound, L-glutamic acid gamma hydrazide, that is a compound that can inhibit enzymes that are dependent on pyridoxal phosphate, like GAD and GABA-T, which are the enzymes responsible for the synthesis and degradation of GABA. When they administered this compound to mice, they found an important elevation of GABA in the brain of the mice, meaning that this compound selectively inhibited GABA and the degradation of GABA. This is the first paper of many about the regulation of GAD that Ricardo published with his students. Also at the Institute of Biology, Ricardo met Herminia Pasantes, and they worked together for several years. They published several papers together, and they become many good friends, very good friends for a lifetime period. Herminia is currently an emeritus professor at the Institute of Cellular Physiology, where Ricardo worked for more than 40 years. After Ricardo graduated from medicine, he enrolled in a doctorate program in biochemistry at the Faculty of Chemistry, and during his studies he made research stay in Houston, Texas, in the laboratory of Jorge Aguapara, where he continued his work on the regulation of GAD activity and its role in the generation of convulsions. And this work was important because their findings suggested for the first time that the blockade of GABA synthesis independently of the concentration levels of GABA in the brain was a very important factor for the production of convulsions. And that highlighted the importance of GABA synthesis at the synaptic cleft. That was an important discovery during that time. Afterwards, Ricardo was graduated as a doctor in biochemistry in 1969, and I have to mention that he was the first doctor to be graduated in the Faculty of Chemistry at UNAM. Soon after he graduated, he went to the United Kingdom to work with Robert Balash, and there he met Dr. Ferenc Hayos, who was an expert electron microscopist, and together they described the structure of the cerebellar glomerulus in a very nice series of 3 papers. When Ricardo came back from the United Kingdom, he became the head of the Department of Experimental Biology at the Institute of Biology. This department was later became the Centro de Investigaciones en Fisiología Celular, and Ricardo was a co-founder of this center. And then this center became the Institute of Cellular Physiology, where many of us work. And I have to say that Ricardo had a very active participation, and he was a very enthusiastic promoter in the planning of this building that hosted the Neuroscience Division at the Institute of Cellular Physiology. Ricardo did many contributions to science. Throughout a 60-year scientific career, he published more than 200 papers, which received more than 6,000 citations. Among his most important contributions were the relationship between GABA synthesis and brain excitability, which I have already mentioned. He was very interested in testing compounds that were derived from vitamin C to inhibit GAD and showed effect on excitability in the brain. He also contributed to the characterization of two types of GAD activity, of glutamate decarboxylase activity. He also contributed to the description of the mechanisms involved in the release of neurotransmitter, also the relationships between excitability and neurodegeneration. And in the last year, he was more involved in elucidating the mechanisms associated with the pathogenesis of amyotrophic lateral sclerosis. From his early career, he was a person that was very interested in the formation of new students. And his first two students that graduated from the doctorate were Miguel Pérez de la Mora and Elena Sandoval, who also worked on the regulation of glutamate decarboxylase activity. Miguel Pérez de la Mora is currently a senior professor at the Institute of Cellular Physiology, and Elena Sandoval unfortunately died when she was very young, due to a tragic accident. This is how Ricardo looked like around 1978, at least in the eyes of Manuel Covarrubias, who is the author of this cartoon. And thank you, Manuel, for providing us this nice drawing. And according to what I have been told, Manuel used to draw these cartoons from the people that were working in the lab of Ricardo at that time, and Ricardo really enjoyed these cartoons. Manuel Covarrubias worked with Ricardo. He was also one of his earliest students. And Manuel worked on the isolated synaptic terminals, and he fused these synaptic terminals with liposomes that were loaded with this metallochromic dye that interacts with calcium, and in this way Manuel could follow the changes in the intrasynaptosomal calcium concentration in different conditions. Florinda Arias also used this preparation, the synaptosomal preparation, and this is a very nice electron microscopy image from a paper that I took from Florinda. And in this preparation, Florinda, together with Maria Sitges, they describe a calcium-independent release of GABA and glutamate. Afterwards, the microdialysis technique was established in Ricardo's lab. Alberto Morales Villagran and myself used this technique to follow the changes in the levels of neurotransmitters in the anesthetized animals. In that moment, what we wanted to do was to block the glutamate transporters and to see if we could get enough glutamate in the synaptic cleft to cause neurodegeneration. And as you can see, this technique was very nice. We could see really the accumulation of glutamate and aspartate in the synaptic cleft, but we didn't observe any degeneration. Later on, Laura Medina and Alberto Morales Villagran coupled this technique with the electrical recording. So they could monitor at the same time the release of neurotransmitters and also record electrical activity in the cerebral cortex. Other students work in other models in vitro, and this is the case of Ivan Velasco, who is a professor at the Institute of Cellular Physiology. And he used neuronal primary cortical cultures as a model of generation and also to test the neurotoxicity of several compounds and the protective properties of some compounds, in this case, gangliosides. Fernando Peña is currently a professor at the Institute of Neurobiology in Querétaro, and he also was interested in the correlation of excitability, GABAergic and glutamatergic neurotransmitter and neurodegeneration. Then Ricardo's lab moved to the spinal cord, and they established a model of chronic neurodegeneration of the motor neurons induced by the chronic infusion of AMPA. And Juan Carlos Corona was one of the first students using this model, and he could induce the death of motor neurons and study its consequences in this model. Luis Tovar Romo and Angelica Cepeda also used this model, and they tested the effect of vascular endothelial growth factor to prevent the death of motor neurons and also the paralysis that was caused in the rats. These are more recent papers of more recent students of Ricardo's lab, who also work in the model of the spinal cord and made other kind of studies. And this is Uri Ramirez Carquin, Rafael Lazo, and Citlali Nexavalrios. Ricardo also did editorial work. He was a member of the editorial board of the most important journals in neurochemistry, Neurochemical Research, Journal of Neurochemistry, and Neurochemistry International, and he was the executive editor of Neurochemistry International for several years. He was also a member of scientific societies, national and international societies, as the Mexican Society for the Mexican Society for Research, and also the Mexican Society of Biochemistry, who this branch of neurobiology belongs to. And he also was a member of the International Society for Neurochemistry and the American Society for Neurochemistry. He was a member of the council of the ISM for several years, and he also participated in the organization of several meetings of this society. This is a very short list of a very long list of all the awards and distinctions that received Ricardo during his life. I'm not going to read them, but I will show you the most important. And one of them is the Premio Universidad Nacional, or the National Prize of the National University Prize in Mexico. And I have to say that this is one of the most important distinctions for scientists at UNAM. And it's important to say that he was the first scientist in all UNAM to receive this award. He also was an emeritus professor at UNAM and also a national emeritus professor. And he also received an award that is given by the City of Mexico to outstanding scientists in basic science. And Ricardo also was awarded with this prize. And the most important probably is the National Prize in Science and Arts that he received in 2002, and it's the most important distinction probably for scientists in our country. So he received that as well. This is also a recognition for his 50 years of uninterrupted work at UNAM. And I have to say that he was about to receive the 60 years recognition of uninterrupted work at UNAM. He also was distinguished in some other ways. For example, in 1998, a special issue of the journal named Neurobiology, which is a journal of Hungarian Academy of Science was dedicated to Ricardo, and this was organized by his friend Beren Hayos and also Graciela Mesa. And this issue appeared 1 year after he was distinguished as an emeritus professor and after a symposium that took place at the Institute of Cellular Physiology. In 2008, he also was distinguished by a special number dedicated to Ricardo, this time in neurochemical research. And this number was edited by Florinda Arias and included 25 papers written by his ex-students and friends. Some of them are listed here. And probably the last distinction that Ricardo received was the organization of this symposium by the Faculty of Chemistry and also the Institute of Cellular Physiology, and Ivan Velasco was the organizer of this symposium, named Chemical Transmission, and we have very distinguished professors participating in this symposium, like Thomas Sufcoff, who received the Nobel Prize, also Manuel Covarrubias, Arturo Alvarez Buya, Karl Kottmann, and among others. Ricardo made very contributions to education. He was a professor of biochemistry in the Faculty of Medicine for 18 years, and he was also a very enthusiastic promoter of the creation of the bachelor, master's, and doctorate program in basic biomedical science at UNAM, and he led this program for several years. And of course, he was also a professor of specialized courses of different graduate programs, and lately also a professor of several courses of the bachelor in neuroscience. This is a list of some of the students that were mentored by Ricardo at the different levels, and I have to say that most of these people are currently doing research either in Mexico or in other places, in other countries. And this is something that Ricardo used to say, and I'm sorry I have to read it in Spanish. Una de las mayores satisfacciones es ver que en el laboratorio donde se elabora se han formado jóvenes, ahora investigadores independientes. Así se contribuye a lo que más se requiere en este país, formación de científicos. And he truly believed that we could contribute, or we can contribute to the progress of our country by the formation of scientists. This is just several photos with many of his students. Then I have to say that Ricardo, his interests were not limited to science. He also was interested in bioethics, and together with his friend Ruben Lisker, he was the co-founder of the College of Bioethics, and he was the president of this college for some years. And he was really very active in the participation in different debates about bioethics subjects like the legalization of marijuana, the legalization of abortion, and things like that. And he wrote many articles in newspapers and also books about that. I have to say that Ricardo was a very sensitive man. He loved classical music, literature, and poetry. Here is a photograph of Ricardo sitting with Fernando Pessoa, his favorite poet. Here is Ricardo standing with Bruckner, and here he's playing the guitar. He liked also to play music. Ricardo was the loving father of two children, Adriana and Ricardo, who are here in the audience, and thank you very much for coming. And he was the grandfather of 4 children, and lately also from these 2 beautiful children. Ricardo married Maria Eugenia Perez Correa. She was very important for him. She's an intelligent woman who emotionally and intellectually supported him for the last 21 years. And here is Ricardo with his brother and sisters. He was the eldest brother of 5 brothers and sisters, and you can see how happy they look in this photograph. This is a picture of many of us that were together with Ricardo during his celebration that, according to my opinion, was a party that he organized by himself to celebrate his birthday, and it was really a very nice party. We enjoyed that very much. And I want to finish with these words that Ricardo said, and they are in Spanish, so I have to read it in Spanish. Como he citado más de una vez en artículos sobre la ciencia, de acuerdo a Ernest Noel en el Cuarteto de Alejandría, la ciencia es la poesía del intelecto y la poesía es la ciencia de los afectos del corazón. Pienso que esta frase es una gran riqueza en más de un sentido. And with this I finish, and thank you very much for your attention, and I hope you have
So moving on with the academic program, we have 2 lectures by 2 alumni of Ricardo Tapia. The first one is going to be by Professor Manuel Covarrubias. I have the honor to introduce him. Professor Manuel Covarrubias is currently a tenured professor at the Department of Neuroscience in the Sidney Kinnell Medical College at Thomas Jefferson University in Philadelphia in the United States, where he is also the co-director of the MD and PhD program. He got his medical degree at the UNAM in 1978, and then he pursued a scientific career under the mentorship of Ricardo Tapia, obtaining his master's in 1979 and his PhD in 1980. Then he did a postdoctoral training at the Max Planck Institute in Dortmund, in Germany in 1981, from 1981 to 1984. And then he did a second postdoctoral training at Washington University in St. Louis, Missouri from 1985 to 1990. Then he became an assistant professor at the Department of Pathology, Anatomy, and Cell Biology in the Jefferson Medical College of Thomas Jefferson University in Philadelphia. Over the past 25 years, he has contributed to the understanding of the molecular physiology and pharmacology of ion channels, with focus on 3 specific areas: the Kv channel-dependent mechanisms of action potential plasticity in the context of brain signaling, the structural basis of Kv mechanisms responsible for the Kv channel specialization, and the structural basis of general anesthetic interactions with the Kv and NAV channels. Channels. More recently, in conjunction with researchers at NIH, Jefferson, and Temple University, Professor Covarrubias launched a new multi-institutional project concerning the structure and function of SARS-CoV-2 viroproteins. The execution of this interest is primarily based on the state-of-art patch lab electrophysiology applied to the birth preparations, including heterologous expression systems, isolated spinal cord, and acutely dissociated neurons. His work is complemented by diverse multidisciplinary approaches, including molecular biology, imaging, protein chemistry, computational biology, structural biology, and optogenetics. Most of his studies involve significant quantitative computational applications, such as detailed analysis of ion channel kinetics and structural modeling, and molecular dynamics simulations conducted by experienced collaborators. Dr. Manuel Covarrubias will be presented tonight at a lecture titled Non-Canonical Gating Control of the Cytoplasmic T1 Domain of the Kv Channels.
Before I start, I would like to thank Rinda, Lourdes, and Van for inviting me to this very, very important event. And what I'm actually going to tell you is actually a story that has a very long history. And it actually began almost 2 decades ago. However, fortunately, just about a year and a half ago, we were very fortunate to work with groups in the UK and Sweden to discover something that really illuminates for the first time with near-atomic resolution certain ideas that we were working on. As I said, long ago, 2 decades ago, I'm going to show you that story and tell you how this story is being resolved. So, but actually, before I continue, it is, I think, I actually have difficulty finding words to express what I really want to say. But it is an honor, a great privilege to actually today have the opportunity to participate in this event. Because, I think to say it in few words, without Ricardo's early influence, I won't be able to enjoy the pleasure of finding things out as much as I have done over the past 4 years of my career. So he played a central and very important role in who I am professionally and what I do and what I like to do. So that's one thing. The other, and of course the most important, as I said, the other is that I would like to give credit to those that actually did the work. As you can imagine, as principal investigators, we often have not much time we spend our time writing grants, writing papers, and doing other things, teaching and administrative responsibilities. So other people actually do the work. So this work was done by a number of people that you see on that slide. Some that are actually currently doing the work, and some that actually just recently joined our efforts. The beautiful structures that I'm going to show you were actually solved by gamma chi. Set up my cursor again. So by gamma chi, which you see here, and the revealing functional mutation studies that I'm going to show you too were actually done in my lab by Tiancheng Liang. Gamma Chi is a member of a group in the UK that is led by Katharina Dur, and some of the really interesting and also revealing simulations, molecular dynamic simulations, were done in the laboratory of Lucy Vanamont, which you see here. So now I would like to answer 2 really, really important questions, because you may ask, why do I care about what I'm going to tell you? And the reason why I care has it's an answer with 2 parts. One is that even 70 years ago, even 70 years after the publication of the seminal papers by Hodgkin and Huxley, we're still fascinated by the mechanisms that underlie the action potential. So beyond the physiological basis, beyond the biophysical basis, what we really want to understand is what are the structural bases of the action potential, of which we know very little, although we still know very little, although a lot has been learned over the past 70 years using a number of approaches that I'm going to present to you, especially with respect to structural biology. So that's one reason why I care. The other reason why I care about this, which is of course super, super important, is that we would like to understand the mechanisms of disease. The channel that you see on this slide, this is actually a cartoon of the channel, this potassium channel that I'm going to be talking about, called KB3.1, has various domains that I'm going to explain in a moment, but in the T1 domain that you see, I highlighted there at the bottom, actually about 50% of the variants that have been found in humans are potentially associated with disease, like epilepsy and other diseases that affect the nervous system. So we think that by the work that I'm going to present you, we are going to learn more about these mechanisms. Where is the pathogenesis at the structural molecular level behind these neurological disorders? So, but actually, before I go into the topic, I know that perhaps many of you are not entirely familiar with the subjects of my talk. So I'm going to give you a little bit of an introduction of what we work on and what these subjects are. So we work on voltage-gated potassium channels. And what you see here on this slide is the phylogeny of these channels, which have basically two properties. They have what is so-called the T1 domain, which is what I'm mainly going to be talking about. And they also have this swapped, domain swapped architecture, which I'm going to explain in a moment, in about 2 slides or so. So these are the subjects. And mainly, I will focus on those that are indicated by the arrows. I'm going to mention a little bit about KB 1.2, which is perhaps the one that is best characterized. Then I'm going to mention I'm going to talk mostly about KB3, so the third family of these channels. And I'm going to historically mention some results that we got actually 2 decades ago on KB4 channels. So to put that in historical perspective and show you why we have these really interesting ideas that began to grow about 2 decades ago. So let's just talk a little bit about the structure. So now we're talking about the domains of a single molecule. We're talking about just one protein, and the domains within that protein, ion channel protein. So these proteins have a subunit that look like what you see here. And basically, if you go from the inside to the outside, or if you go from it's called the N-terminus to the C-terminus of the protein, we can actually define 3 domains. You have the T1 domain, the most N-terminal, followed by the voltage sensing domain, which includes segments S1 to S4, and that's followed by the pore domain, which includes the segments S5, the so-called pore helix, and the S6 segment. Now, this is something that actually already tells quite a bit. As I mentioned, there is a voltage sensing domain and there is a pore domain. But how do they communicate one to the other? Well, it's actually that little short helix that you see here labeled 4-5. This helix actually allows the communication of the voltage sensing domain with the pore domain. And remember, one thing that I have said already a few times is that these channels are voltage gated. What that means is that the membrane potential is the stimulus, changes in the membrane potential is the stimulus that actually gates or allows these channels to do something in our brains. So now this is just one subunit, but they don't actually work like that. They actually 4 of these actually combine to form what is called the tetrameric. Tetrameric meaning 4 subunits coming together to form the actual functional pore. And there you see in 4 different colors the 4 subunits forming that functional KB channel. In this case, the KB 1.2. So then, as I mentioned just a moment ago, these channels have the domain swapped architecture. What that means is that, as you can see at the bottom here, on this picture here, as you can see, one green subunit interacts with the neighboring subunit, like the green interacts with the orange, the orange interacts with the blue, and so on. That's really important too. That means that in order to open this channel and so that the channel can do what it's supposed to do, there is an inter-subunit interaction of one voltage sensing domain with the pore domain of the neighboring subunit. Now, what do we know? So far, this is actually very current. What is the state of the field in terms of how these machines work? And I would like you to think about what I'm going to be telling you about as machines. We're trying to understand how these machines, ion channel machines, actually work. So what do we know currently about the mechanisms that underlie what I was telling you about? Basically, there are 2 non-exclusive mechanisms. One is known as the canonical mechanism, and the other one is the non-canonical mechanism. So what you see here is the canonical mechanism. And I already said a little bit about what is going on this picture. As you can see, there is this segment, what is called the S4. S4 is the actual voltage sensor. This is what senses the changes in membrane potential. And I'm going to show you a movie in a moment where you can actually more graphically, visually see how it works. But basically what you see here is that the voltage sensing domain is going to move up and down depending on how the membrane potential of the neuron changes. And it communicates that to the pore domain, or a very important part of the pore domain, which is shown here in green, through that short helix that I was telling you about, the S4, S5. So whenever that sensor moves out, it pulls on that linker, and that allows the port to expand and open. And again, as I said, I'm going to show you the movie of that in a moment. So you can actually think about this idea as a hand and wrench. The hand turns the wrench, and the channel opens. So thinking in terms of a machine analogy. So but there is also a non-canonical mechanism that was proposed very recently, probably within the last 6 to 8 years. And in that one, which is actually non-exclusive, that means we can have both together, working together to control this machine. In that one, we also assume that the voltage sensor moves out, but instead of just simply pulling on that linker that I was telling you about, it's going to interact, what we call allosterically, because it's at a distance. It's interacting with another important segment, which is the S5 segment. And through that allosteric interaction is also controlling that gate that I was referring to, which is again shown in green here. Now, I would like you to just kind of remember that what I'm going to refer as the gate is the S6T, which stands by S6 tail. So that's kind of the same synonym of the definition of in words, what the gate of these channels is. So we can see that as a rapid pinion. When the S4 moves, then it actually moves the S5, and then that transmits the movement or the conformational change to the port gate that opens the channel. Here's the movie. So let me just explain to you what you're going to see. This is just for simplicity. It's just showing one voltage sensing domain. Remember that I showed you that these channels are tetrameric, so we actually have 4 of those. But just for simplicity, so you can see what's going on, I'm going to show you just one voltage sensing domain and the port domain, which is all shown in yellow. So in the voltage sensing domain, you have this really interesting segment shown in pink, which is, as I said earlier, the S4 segment. The S4 segment characteristically has arginine groups. So these are amino acids that are positively charged. So you can imagine charges can sense changes in memory potential. So these arginines actually are going to sense when the memory potential changes, and they're going to move either up, meaning towards the outside of the cell, to activate the channel, or they're going to move in to actually deactivate the channel. So what you're going to see is that pink segment moving up because the channel is being activated, transmitting that movement to this linker that I kept talking about, the S4S5 linker, and then you're going to see the port actually expanding, which is when the channel is actually open. So you see those arginines crossing what is called as the charge transfer center, pulling on the S4S5 linker, and actually opening. But I think it's actually better to see that from the inside. So I'm going to show that structure that you see there, 90 degrees, so you can actually see it from the inside and see how the port actually expands when the voltage sensor actually moves into the screen. So now we have the same model, just looking at one voltage sensor. And you're going to see the voltage sensor, here in pink again, moving into the screen. And as it moves into the screen and crosses that charge, the charge transfer center, the S4S5 linkers, which you see in blue, and notice that these S4S5 linkers actually kind of form a cuff around the port. Right? So the cuff is going to expand as the voltage sensor actually moves up or into the screen, as I was just saying. So that's how it works, in a way, right? Or to a certain extent, that's how it works. And whether you think about the canonical model that I showed you, or the non-canonical model, that's pretty much very similar mechanism. However, what is kind of intriguing about this is that these models and these studies are actually ignoring a very important part of the channel. Which brings me to the elephant in the room. The elephant in the room is something that I already told you about, the cytoplasmic T1 domain. This region, this very large part of it is almost half of the mass. Half of the mass of the channel is the cytoplasmic T1 domain. So we wanted to know, actually 2 decades ago, what this was doing, because we had certain ideas that I'm going to show you in a moment that suggested that this cytoplasmic T1 domain may actually also contribute to these conformational changes that I was telling you about. And these models that I told you ignore this. In other words, they are structurally incomplete. They don't actually take into consideration this elephant in the room. So, but there are a few things that we already know about this cytoplasmic T1 domain, and we like to call this domain as the multitasking T1 domain of potassium channels, voltage-gated potassium channels. It is known, and actually this is what I did at Washington University, that they actually contribute to the specificity of subunit assembly. They also contribute to the docking of auxiliary subunits, and towards the end, I'm going to show you some images of that. They also play some role in subcellular targeting, whether the channels go to the axons of the synapses, or whether the channels go to the somatodendritic region. That actually depends also on the T1 domain. So the question we were asking, can they actually do one more thing? Can they actually contribute to gating control? So, and this is where I go back 20 years to show you what we were thinking. So, this is the X-ray crystallography of the structure of this domain using X-ray crystallography. This is just the T1 domain. We don't see the rest of the channel. The rest of the channel is outside of the screen. At that time, the structure of the so-called transmembrane segments of the channels were not known at all. We didn't have any idea of how they looked like in detail. But we knew how the T1 domain looked like. And what you see here is the structure of the T1 domain, this cytoplasmic domain, in KB 4.2. So this is another member of this family that I showed you, the phylogenetic tree. And you can see that segment that I'm going to keep talking about this alpha-6 helix all over and over again over the rest of the talk. So this is what really we care about, alpha-6. And as you can see, alpha-6 is kind of pointing up. And remember that this is a tetramer, so we have this pointing up and this pointing up. Right? So, and we thought at that time, if this is pointing up, right, and the rest of the channel is sitting here as a hat on my head, right, it must actually talk to that. It must tell something to the rest of the channel. And we say, well, maybe it's telling something about gating. And what made that even more interesting is that another structure that was available at that time was that of KB 1.2, which is kind of the stereotypical, the archetypical potassium channel, KB 1.2. And as you can see, the alpha-6 helix is actually not pointing up, it's actually pointing down. So what I'm going to tell you may not actually apply to KB 1.2. We're still speculating and thinking about hypotheses to explain this. But what I'm going to tell you probably applies not to KB 1.2, but it applies certainly to KB 3, as I'm going to convince you of, and it applies to KB 4, because I'm going to show you data about KB 4, and we speculate that it may apply also to KB 2. And I'm going to actually, in a couple of slides, even convince you more about that hypothesis. That is not KB 1.2 or KB 1, but it's all KB 2s, KB 3s, and KB 4s. So this is very intriguing to see these two differences. Right? That one is pointing up, the other one is pointing down. Potential is actually 0. So the structure that I show you is actually at 0 millivolts. When the membrane potential is changed, we actually use molecular dynamics simulations. However, this is something that we really want to do. We want to capture the channel in different states. This is actually just one state, one snapshot of the channel, in what appears to be actually an open conformation. But we don't know how it looks when it's at rest. We don't know how it looks when it's inactivated. So we're going to use these similar approaches to the one I just described, using systems to cross-link different parts of the channel and then lock in the channel in different conformations. And that will be really exciting to do. Thank you for the question.
Thank you. Well, if there's no more questions, please join me in thanking Dr. Morales again. So next we have a lecture by Dr. Laura Medina Ceja, also a former student of Ricardo Tapia. Laura Medina Ceja is a biologist from the University of Guadalajara. She completed a master's degree in basic biomedical research and a doctorate in biomedical sciences in the Institute of Cell Physiology at UNAM under the mentorship of Ricardo Tapia. She did a postdoctoral training in epilepsy at the Department of Neurology at the David Geffen School of Medicine in the University of California, Los Angeles. She is currently the head of the Neurophysiology Laboratory and professor and senior researcher. She's a member of the National System of Investigators Level 2. She has 30 years of experience in experimental epilepsy. She's also a member of the American Epilepsy Society, the American Society for Neurosciences, and the International Brain Research Organization. Dr. Medina Ceja will present tonight the seminar entitled Electrophysiological Biomarkers of Epileptogenesis, new insights about its utility and the neurobiology of the hippocampus and clinics.
Thank you. First, I want to express my gratitude to the organizing committee of this for neurobiology meeting, and special to Dr. Ivan Velasco and shares Dr. Florinda Arias and Dr. Lourdes Maciel for this special invitation for this symposium about the legacy of the scientific legacy of late Professor Ricardo Tapia. And I want to talk about my mentor, Dr. Ricardo Tapia, as a legacy of discipline, honesty, work, and resilience during my formative experience in his laboratory. So he always inspired me to get real objectives about my academic formation and work with bioethical principles to permeate all my academic activity and, of course, my life. This attitude in the life not always gratify people, but I think this is a rare condition that sum to the justice and equity in our research relate. So thank you so much, Dr. Ricardo Tapia, for your time, your opinions, advice, and sporadic scolding that follow me, that allow me to get improvement opportunities to be a better researcher, professor, and guide to my students. And your legacy will remain in your students and indirectly in the students of your students. So thank you so much, Dr. Ricardo Tapia. Well, my talk is about the electrophysiological biomarkers of epileptogenesis and how these biomarkers give us new insights about its utility in the neurobiology of the hippocampus, capable to induce them and clinic. For the items to review in this talk are the basic concept in order to get into the subject, contribution of the electrophysiological biomarkers, fast ripple, in the neurobiology of the hippocampus, capable to induce these fast ripples, and the relevance of fast ripple in clinic. So first we need to know that epilepsy is a brain disease, a chronic disease, that defined by any of the following conditions according to the institution that dictates all the terms about epilepsy, that is the International League Against Epilepsy. And they told us about this following condition in a person who take an epilepsy condition. Is at least 2 unprovoked reflex seizures occurring 24 hours, or 1 unprovoked seizures, and probability of further seizures similar to the general recurrence risk, that is 60% after 2 unprovoked seizures occurring at the next 10 years, and when we have a diagnosis of epilepsy syndrome. Well, to know about the relevance of the epilepsy as a health global problem, the studies in the epidemiology of epilepsy estimates that epilepsy is in the 70 million people worldwide. And the few studies in Latin America and Mexico tell us that there is an estimation of 5 million to 1.5 to 2 million people with epilepsy. So this international institution classified the seizure types in three principal types of seizures according to its basic version, as I showed you: the focal onset, generalized onset, and non-onset. And if these three seizure types, the first one have two subdivisions. It's aware, or that it's compromised or not, and with motor and non-motor manifestations. So we focus in this, we focus in this first type of focal onset because temporal lobe epilepsy is the main form of localized epilepsy. And because in this disease was observed for the first time the pathological high-frequency oscillations or the biomarkers of epileptogenicity that are very important for the epilepsy. And it's a highly drug-resistant disease that is generally treated using surgical intervention. Moreover, in animal models that simulate this disease, also were observed pathological high-frequency oscillation, these important electrophysiological biomarkers, and even they were found before they were found in patients. So in these animal models of epilepsy, we can obtain valuable information about the mechanisms of initiation and of epilepsy, as well as the action mechanisms of seizures, drugs, and resistance, as well as the presence of molecular and electrophysiological biomarkers of epilepsy. And in both patients with temporal lobe epilepsy and animal models, there is a complex process that we call epileptogenesis that includes a sequential pathological events that trigger this disease. So it can angle define epileptogenesis as the development and extension of tissue capable to generate spontaneous seizures, and resulting in the development of an epileptic condition, or well, in the progression of this epilepsy condition after it is established. So this pathological process begins with a primary injury that could be an impact factor or impulse force or genetic or metabolic alteration, as well as infection agents that can trigger these pathological events. So we have here the necrosis, as well axonal injury, gliosis, apoptosis, until the presence of chronic neurodegenerations during the different times according to the model of epilepsy. So the principal proof about this pathological condition is the hippocampal sclerosis, that is the most frequent pathophysiological evidence of temporal lobe epilepsy, both in patients and in animals with this disease. So here I show you some image in which we can find neural loss in the delta gyrus and the hippocampal sclerosis and the delta gyrus. As well, in the image of animal models, we can observe this pathophysiological structure that is the hippocampal sclerosis, is protein in the dental gyrus that correlate with a high immunoreactivity to the microglia. And why this pathophysiological evidence is found in this structure in particular? Well, because hippocampus exhibits a high degree of neuroplasticity and have a great control of the temporal dynamics of its circuits to encode information. And these properties confer to this structure an intrinsic epileptogenicity. So, in addition, when we have an epileptic condition, there is a loss in the excitatory and inhibitory control in this structure, and in particular in the third synaptic circuit, that is the principal circuit in this structure. So here I show you the third synaptic circuit, the canonical and non-canonical connection in the third synaptic circuit, and begins with the entorhinal cortex that receive inputs of different subcortical regions that reach the hippocampal structures, and this entorhinal cortex send a principal projection to the dental gyrus through the peripheral pathway, and the ganglion cells of the dental gyrus send an important connection to the pyramidal cells of CA3 through the muscle fibers. And in this area of the hippocampus, we observe abundant recurrent connection and a principal connection to the pyramidal cells of CA1 through the Schaefer collaterals. And this CA1 area of the hippocampus send in information or connections to the cerebellum and the entorhinal cortex, completing this trisynaptic circuit. But recently, it is described other non-canonical connection in this principal region of the trisynaptic circuit, that is the CA3 back projection to the dental gyrus, and the cerebellar back projection to the CA1. And this is our very important connection that we retrieved in our talk. So in this trisynaptic circuit, in the epilepsy or in the epileptogenesis condition, we observe morphological and genetic alteration like the hippocampal sclerosis, and neurochemical alterations with an increase in the excitatory over the inhibitory neurotransmission. Also, we have electrophysiological alterations and different patterns of epileptic form activity, as well as the electrophysiological biomarkers of this complex process of epileptogenesis. All these alterations have profound effects, and they can get pathological neural networks in this trisynaptic circuit. So the principal electrophysiological alteration that we found in these structures is the pathological high-frequency oscillations. And they were found in models of temporal lobe epilepsy and patients. And we have two types of these oscillations, ripples and fast ripples. Ripples have a frequency of 100 to 250 hertz, and fast ripples have a frequency of 250 to 600 hertz. They are found in the gyrus, CA3, and CA1 of the hippocampus. And here I show you some representative ripples and fast ripple superimposed to an interictal spike, or superimposed to a slow wave. And even we can observe the two ripples and fast ripple in the same slow wave, beginning with the fast ripple activity and then with ripple activity. And why they are important biomarkers of electrogenesis, and in particular fast ripple? Well, because fast ripple were observed before and during seizures, and because fast ripple also were found in seizure onset zone in the focal region of the tissue capable to generate spontaneous seizures, and also because there is a relation with more fast ripple in the presence of more seizures. In addition, fast ripple were reported in quiet wakefulness and slow wake sleep states, and they are the response of cluster of pathological associated neurons with a volume of 1 cubic millimeter of tissue. So with these basic concepts, now I want to show you some of our principal findings about fast ripple in epileptogenesis in the hippocampus. For this purpose, we use in vivo model of epilepsy. Animals were administered with pilocarpine, and after 4 to 6 months, the animals have spontaneous and recurrent seizures, and then with electrophysiological techniques, we observe fast ripple in these animals and the hippocampus. So with this model, now we want to investigate about the serotonin modulation of fast ripple activity, because the serotonin transmission is very important and is involved in epilepsy, and because we observe a high occurrence of fast ripple activity during the slow wake sleep period, during which we observe a decrease in the levels of serotonin. So we take on the task to investigate about these fast ripples and their modulation through this serotonin neurotransmission. For this purpose, we use the citalopram. The citalopram is a blocker or serotonin reuptake, so we can increase the cellular concentration of serotonin. And when we administrate citalopram, we observe a clear decrease in the occurrence of spontaneous fast ripples, as well in the number of oscillation cycles per fast ripple event, and in the average fast ripple frequency. And here I show you some representative intracranial EEG traces in different regions of the hippocampus with the fast ripple activity, and when we administrate citalopram, we observe a clear decrease in the number of oscillation per fast ripple event. Also, when we withdraw the citalopram for the Kerr-Ringer solution, we observe a reestablishment of this pathological oscillation over the time. So with these interesting results, we now want to know about the principal serotonin receptors involved in epilepsy and maybe in the fast ripple generation. So we use these antagonists for the 5-HT1A receptor and for the 5-HT2AC receptors, weight 100 and 145, and 3 times 7 in this same model of epilepsy. And when we administrate this antagonist, we observe a clear increase in the fast ripple events, also increase in the duration and in the amplitude of fast ripple events. So here I show you some representative intracranial EEG recordings before and at different times after the administration of this antagonist. In addition, we carry out other experiments in which we evaluate the immunoreactivity of the principal serotonin receptors. So we obviously choose the 5-HT1, 2, and HT7 that are involved in epilepsy and also in fast revolt. And we analyze the three principal regions of the trisynaptic circuit, and we observe a clear decrease in the immunoreactivity of 1A and 2 receptors in the dental gyrus, and a decrease in the 1A and 7 receptors in the CA3 region of the hippocampus. In these same animals, we evaluate the extracellular concentration of serotonin and its metabolite 5-hydroxyacetic acid, and we observe a decrease in these metabolites in the hippocampus epileptic rats with fast revolt. Well, with all this data, now we can suggest that there is an important modulation of serotonin in the fast revolt generation. So we observe different alterations in the neuromodulation of serotonin system under the epileptogenesis, in particular in dental gyrus and CA3, that it is a very important factor to promote the occurrence of fast revolt in this structure. So the alteration in the projection, in the serotonin projection coming from the raphe nucleus to the GABA interneurons in CA3 and dental gyrus involves a decrease in the serotonin and its metabolite levels, also in the decrease in the immunoreactivity to the principal serotonin receptors, and in particular in the HT1 receptor. And all these alterations we suggest that are very important factors that can generate fast revolt generation in the hippocampus. And next, we evaluate the glutamate control of fast revolt activity, considering that the principal neurons of these three regions of the trisynaptic circuit are involved in fast revolt activity. And because fast revolt are the result of the spike population bursts of the neuropathological interconnected neurons of this structure in the trisynaptic circuit. So we use two antagonists, the CNQX and MPPP. They are antagonists of the AMPA receptor and metabotropic glutamate receptor 5, and we observe a clear decrease in the fast revolt events with both antagonists. So here I show you some representative intracranial EEG traces before and at different times after this administration of this antagonist. And in addition, we evaluate the immunoreactivity of these glutamate receptors, and we include the NMDA receptor, and we observe a clear increase in the immunoreactivity of the AMPA, NMDA, and mGlu5 receptors, in particular in CA3, but as an interesting result, we have an increase in the AMPA receptors in the three regions of the trisynaptic circuit. And also we correlate these findings with the reactive gliosis, with the immunoreactivity of GFAP. In the three regions, we observe this reactive gliosis and a loss, neural loss in the C3 and dental gyrus with the new immunoreactivity. And also we evaluate the extracellular concentration of glutamate in these animals, but we didn't find any change in glutamate levels. Maybe because we evaluate at a specific time in the tissue samples, so if we can use another methodology that we can observe online levels, different changes in the levels of glutamate, and we correlate with fast ripple activity. We have better findings about this. So with these results, now we can suggest that the hippocampus that capable to generate fast ripple activity, it is very important that maybe overactivation of this AMPA and kinase receptor and these glutamatergic cells of the three principal sites or areas of the trisynaptic circuit that correlate with a high reticulosis in these three areas of the trisynaptic circuit that are very important to generate these fast ripples. And in previous studies in my lab, we observed we evaluate the electrical synapses and their participation in the epilepsy in acute seizures induced by foraminopyridine, and we obtained very interesting results about the participation of the electrical synapses or gap junction in the epilepsy, because we observed a clear decrease in the epileptic activity, also in the convulsion seizures. So in other studies, in vitro and in vivo, they have a relevant participation about these electrical synapses in epilepsy, and in silico studies have demonstrated an important contribution of the electrical coupling in the neurons to activate and generate gamma and fast ripple activity. So for this purpose, we use two blockers of connexins. Connexins are structural proteins of the gap junctions or electrosynapses. So when we use the carbenoxolone, that is a specific blocker of connexins, or we use the quinine, that is a dose-dependent blocker of connexin 36, we observe a significant decrease in the number of fast ripple in different regions of the hippocampus, as well in the number of oscillation per fast ripple event during and at different times after the carbenoxolone or quinine administration. So here I show you some electrophysiological traces about this before and at different times after the blockers administration. And with this result, now we can suggest an important contribution of the electric synapses or gap junctions, and in particular in this region, in the dental layers to CA3 through the muscle fibers, and because some studies have found electric synapses in this region, and in particular these synapses that they participate in the synchronization of electrical activity. So we suggest that an overexpression of these mechanisms can generate fast ripple in the trisynaptic circuit of the hippocampus. And finally, considering that we find our principal alterations in the fast ripple activity in the dental gyrus and CA3, and most of the occurrence of fast ripple that we observe are in the CA1 region, we take on the task to investigate the relationship among these areas of the trisynaptic circuit in the fast ripple activity generated in CA1. So here I show you some spike probability distribution of the activity in the three regions of the hippocampus. We can observe synchronization in the peak-to-peak, maximum peak-to-peak amplitude of this activity, and also a quasi-normal And in the panel, we can observe the cumulative spike probability of this region and how the CA3-1 is the first region that synchronized and have an increase in the cumulative spike probability before the fast ripple event, and the close normal distribution in the CA1. With these results, now we can suggest that the CA3-1 is an important area for the initiation of this fast ripple activity. Then we want to know about the fast ripple synchronization with other frequency bands. So we use the theta coherence and gamma coherence, and we observe a synchronization of the theta band with the fast ripple activity, while the gamma coherence remains fluctuating during the experiment. Then we make a statistical comparison in the two theta bands and in the three pairs of connection before and after the fast ripple event, and we observe an important and significant results before the fast ripple event and in the three pairs of connections. And to evaluate the directionality of this connection in the three regions of the trisynaptic circuit, we make a Ranger causality test in this fast ripple events, and also a meta-analysis to determine the overall p-value 200 milliseconds before and 200 milliseconds after the fast ripple event. And this is the results in this table where you can find here that the connection or the principal connection before the fast ripple event are the CA3 to delta gyrus back projection. So this is a preponderant involvement of this back projection before the fast ripple event, and then we observe after the fast ripple event the canonical connections that are in the trisynaptic circuit, the delta gyrus to CA3 through the master fibers, and the CA3 to CA1 through these Schaefer collaterals. So here in this image you can observe this preponderant connection before and after the fast ripple activity, and we can observe this important connection, the C3 back projection to the delta gyrus before the fast ripple event. So when we evaluate the unit activity density in these three regions, we observe a clear and important contribution of CA3, maybe as a hub, as a causal hub in the generation of this fast ripple, and the delta gyrus as a support site for this generation of fast ripple activity. Well, with all this data, now we can contribute to the neurobiology of the hippocampus capable to induce fast ripple. And this is a summary of the principal findings in this trisynaptic circuit. The important reactive gliosis in the three principal regions of the trisynaptic circuit, with the neural loss in the delta gyrus, as was supported by others reports. Also the important serotonin modulation of fast ripple with the damage of alteration in the inhibition of this projection coming from the reflex nucleus to the hippocampus, which includes a decrease in serotonin levels and its metabolites, and a decrease in the immunoreactivity of principal serotonin receptor, and in particular in 5-HT1 receptor. Also, the importance of maybe the overactivation of ampine kinase receptor in the two principal region of the first synaptic circuit, and maybe an involvement of metabotropic glutamate R5 receptor in CA3, and how the electric synapses can modulate this fast ripple, maybe with the overactivation of the mechanisms of these electric synapses that synchronize electric activity. And these pathological conditions in the trisynaptic circuit form a reverberant excitatory circuit that it is driven by this CA3 back projection to the entire gyrus that can generate fast ripple in this hippocampus with epileptogenesis. So finally, I want to talk about the relevance of these electrophysiological biomarkers in clinic, because fast ripple ripples and fast ripples both are found in temporal lobe patients, and because these fast ripples are very good biomarkers of epileptogenesis. So, fast ripple activity now has been used as a surgical reference to detect candidate areas for resection with resultant seizure-free outcomes in patients. And I show you the support vector machine that is used to identify pathological high-frequency oscillation and elaborate a classification of this pathological oscillation, and it is used in the future surgical neurologist. This is one of the several studies in which fast ripple and ripple are used to determine the area for resection in patients. So we have the spatial distribution of these fast ripples in specific electrodes, and in the yellow line you are observe the margin of this area for resection, and in this patient with free seizure outcomes of this patient follow up for 2 years. And this is the principal institution that used this high-frequency oscillation as a reference to detect the area for resection in patients with important results in patients with free seizure outcomes in these patients. In different countries in the world, we have in Canada, USA, and Europe, and Switzerland, Germany, and Netherlands, and in China and South Korea. Then, hopefully, one day in Mexico, we can use this pathological high-frequency oscillation as a biomarkers of epileptogenesis, and to obtain areas for resection in patients with this temporal lobe epilepsy. And hopefully we have important results as in the other countries. So now I want to show you my work team in my lab. It's Dr. Kenia Pardo, Dr. Consuelo Ventura, Dr. Frida Villalpando, and obviously the important and valuable contribution of our students in the different programs. So thank you for your attention, and thank you very much again, Dr. Ricardo Tapia.
So we have time for one questions from the audience.
Thank you, Laura, for this nice talk. I have two questions.
Well, the first one is whether it is known something about the levels of serotonin in patients with epilepsy, and also maybe in post-mortem analysis about the abundance of serotonin receptors.
Yes, there are some studies in which the cellular concentration of serotonin was evaluated in patients with temporal epilepsy by microdialysis, and they found a decrease in the serotonin levels. Also, there are some studies in which the immunoreactivity for these principal serotonin receptors were evaluated, and they found, in particular, a decrease in the immunoreactivity of the 5-HT1A receptor.
Okay. And the other one is, what do you think is doing the reactive astroglia in the development of these fast ripples?
Well, this pathophysiological evidence, that is the reactive gliosis in these two regions, was found in other different types of epilepsies. The gliosis is more involved in the acute events during an epileptogenesis, during this complex process that generates another types of pathological events that are relevant for the epilepsy, for example, the neurodegenerations. But really, we didn't know about what is the relevance of this gliosis in, for example, fast ripple activity. So this is a very interesting topic that maybe we can take on the task in the future in our lab.
Thank you very much.
I have a question. It is known that generation of fast network oscillations in the hippocampus relies on GABAergic interneurons. So I was wondering if you know if there are some qualitative or quantitative alterations in specific subsets of GABAergic interneurons in the hippocampus of the epileptic brain.
Yes, there are other studies in the laboratory of Dr. Anatol Ragin, and he studied some aspect of this, about this observation, and they found that there is a very small volume of pathological tissue that generates this fast ripple, and the neurons that are involved in these high-frequency oscillations are glutamatergic and the GABAergic interneurons that are associated with VIP, for example. And I think this is the more relevant studies about your observation.
Thank you.
You're welcome.
Thank you very much, Dr. Medina Ceja. And for finishing off this session, we would like to thank again the family of Ricardo Tapia for being here tonight. And we would like to invite all the audience to give a standing ovation in the memory of Ricardo Tapia.